Welcome to our lecture on human louse control. Imagine hosting a party where tiny, uninvited guests arrive, hide in the carpets, and constantly bite you. This is exactly what happens when human lice infest a person's hair or clothing. Today, we're going to explore not just how to get rid of these stubborn parasites, but the rigorous science and mathematics used to ensure our treatments actually work. Let us dive in. To begin, we must understand our enemy. Human lice are tiny, wingless insects that have evolved to live exclusively on us, relying entirely on our blood to survive. Getting rid of them is not as simple as taking a normal shower. Because they are so well adapted to the human body, removing them requires a targeted, scientific eviction strategy. We cannot just wash them away. We have to systematically break their life cycle to ensure they do not return. This systematic approach is what we call human louse control. Now that we know what they are, let us look at the different types. There are 3 specific types of lice that infest humans: the head louse, the body louse, and the pubic louse. While head and pubic lice are primarily highly irritating nuisances that cause severe itching and social stigma, the body louse is a completely different threat. It is a dangerous biological vector. Historically, body lice have transmitted deadly diseases like epidemic typhus, trench fever, and louse-borne relapsing fever, wiping out millions during times of war and famine. This makes control a critical public health measure. Because the stakes are so high, how do we actually fight them? Human louse control relies on a multifaceted approach. This process involves a combination of chemical treatments, known as pediculicides, mechanical removal using specialized combs, and environmental management, like washing clothes at high temperatures. The primary goal across all these methods is to completely break the life cycle of the louse. This means ensuring that both the living adults and their unhatched eggs, which are called nits, are completely destroyed. Modern strategies must be highly adaptive because lice have developed resistance to many common chemicals over the years. Let us visualize this multifaceted approach with a diagram. On the screen, you can see the 3 pillars of louse control we just discussed. On the left, we have chemical control, represented by the pediculicide lotion applied to the hair. In the middle is mechanical control, showing a fine-toothed nit comb physically removing eggs from the hair shaft. On the right, environmental control is shown by the high-temperature washing machine for clothing and bedding. Notice how the arrows from all 3 distinct methods converge at the bottom onto a single goal: total eradication. Relying on just one method often fails, but combining all 3 effectively breaks the rapid reproductive cycle. With these control methods in mind, we face a new challenge. How do we know if a new chemical treatment actually works? When scientists develop a new chemical shampoo or lotion to kill lice, they need a mathematical way to prove its efficacy. They cannot just look at a patient and guess that fewer lice are present. Furthermore, they must account for the fact that some lice might have died naturally during the testing period. To find the true success rate of the chemical, scientists compare a treated group of patients to an untreated control group, using a standard calculation. This brings us to the mathematical standard used by scientists, known as Abbott's formula. Conceptually, this formula isolates the exact percentage of lice that were killed specifically by the chemical intervention, filtering out natural deaths. Let us look at the symbols. E represents the efficacy of the pediculicide, expressed as a percentage. C stands for the number of surviving lice in the untreated control group after a specific time period. T represents the number of surviving lice in the group that received the chemical treatment. Physically, if a treatment is highly effective, the number of survivors in the treated group, T, will be very small. As T decreases, the overall efficacy, E, will increase and approach 100%. Now that we have our formula, let us see this with a concrete example. A public health team is testing a new permethrin-based lotion for head lice. We are given that the untreated control group has 200 surviving lice after 24 hours. The group treated with the new lotion has only 10 surviving lice after the same period. We need to calculate the efficacy to see if this treatment is viable for public use. We chose Abbott's formula because it perfectly compares these two groups. We substitute 200 for C and 10 for T. Subtracting 10 from 200 gives us 190. Dividing by 200 and multiplying by 100 gives us our final answer. The new lotion has a 95% efficacy rate, meaning it is highly successful and likely to be approved. So, we have successfully used this formula to find the efficacy. But to truly understand this mathematical proof required by health agencies, let us break down why this specific fraction works. We start by looking at the numerator, C minus T. When we substituted our clinical trial values, we calculated 200 minus 10, which equals 190. The mathematical reasoning here is crucial. By subtracting the treated survivors from the controlled survivors, we are isolating the exact quantity of lice that were killed specifically by the chemical. This step ensures that our final percentage reflects only the chemical's true killing power, completely excluding any lice that might have died from natural causes during the 24-hour period. Continuing with our derivation, we now look at the entire fraction. We take that isolated numerator of 190 and divide it by the control group size, which is 200. The intuition here is that we must normalize the number of chemically killed lice against the baseline population that would have survived without any intervention. This gives us a decimal ratio. Finally, we multiply by 100 to convert this ratio into a standard percentage. This mathematical proof is exactly what health agencies require. It definitively proves that the drug has a 95% success rate, justifying its approval for mass distribution to control outbreaks in schools and communities. Now that we know how to prove a treatment works, we must understand why we need to apply it immediately. Lice reproduce rapidly and continuously when they are in favorable conditions on a human host. Waiting to treat an infestation is a terrible idea because the population can quickly spiral out of control. As the number of lice increases, it drastically elevates the patient's discomfort through severe itching. More importantly, a massive increase in the louse population significantly raises the risk of transmitting deadly diseases to others. To understand this absolute urgency, we need to model how fast they actually multiply. To model this rapid reproduction, scientists use the standard exponential growth formula. Conceptually, this formula shows how a population grows continuously over time when resources, like human blood, are unlimited. Let us define the variables. N sub t represents the final population size of the lice after a given time. N sub 0 stands for the initial population size at the start of the observation. The variable r is the intrinsic daily growth rate, and t is the time elapsed in days. Finally, e is a mathematical constant, approximately equal to 2.718. Physically, if the growth rate r, or the time t, increases, the final population, n sub t, will explode exponentially. Let us apply this formula to a real-world scenario to see the danger of waiting. We are given that a child is infested with an initial population of just 10 adult female lice. The continuous daily growth rate is 0.15 due to favorable conditions. We're asked to find the population after 20 days if no control measures are taken. We choose the exponential growth formula because lice reproduce continuously. We substitute 10 for n sub 0, 0.15 for r, and 20 for t. When we calculate this out, we find that the final population is approximately 201 adult lice. This physically demonstrates how a barely noticeable infestation becomes severe in less than 3 weeks. Before we conclude, let us break down the mathematical reasoning behind that massive population explosion. We start by looking at the exponent in our equation, which is the daily rate of 0.15 multiplied by 20 days. When we multiply these together, we get a clean value of 3. Our equation now simplifies to n sub t equals 10 times e to the power of 3. The intuition here is that we are consolidating the daily growth rate and the total time into a single powerful growth factor. This shows exactly how the passage of time directly amplifies the reproductive rate. Now for the final steps of our derivation. We evaluate the exponential term, e to the power of 3. Since e is roughly 2.718, cubing it gives us approximately 20.085. This number means the population will grow to 20 times its original size. Finally, we multiply this factor by our initial population of 10, giving us approximately 201 lice. This step-by-step breakdown mathematically proves why immediate louse control is so critical. It is not just a slow increase, it is an exponential explosion that must be stopped early. We have covered a lot of ground today, so let us review our key takeaways. First, human louse control is a multifaceted approach requiring chemical, mechanical, and environmental methods to completely break the parasite's life cycle. Second, we learned that while head lice are a nuisance, body lice are dangerous vectors for severe, life-threatening diseases. Third, we saw how Abbott's formula mathematically proves the true efficacy of chemical treatments, ensuring public health safety. Finally, our exponential growth models highlighted the absolute urgency of immediate intervention. By understanding both the biology and the mathematics, we can effectively defend against these outbreaks. Great job following along.